Good day, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It is 7:07:37 seconds a.m. Eastern Standard Time. Science News. Test detects protein associated with Alzheimer's and CTE. Findings could lead to early diagnosis, better treatment studies. Date December 20th, 2018. Source National Institute of Allergy and Infectious Disease. Summary. An ultrasensitive test has been developed that detects a corrupted protein associated with Alzheimer's disease and chronic traumatic encephalopathy, CTE, a condition found in athletes, military veterans, and others with a history of repetitive brain trauma. This advance could lead to early diagnosis of these conditions and open new research into how they originate. The test is extremely sensitive. For example, if a pinhead-sized example of brain tissue from an Alzheimer's patient were pulverized and diluted into 1,000 gallons of liquid, the test still could detect tau seeds in pinhead-sized volume in that dilution. The test is called ADRTQUIC, Alzheimer's disease, real-time quaking-induced conversion. Scientists at the National Institute of Allergy and Infectious Disease developed RTQUIC about a decade ago to detect Creutzfeldt-Jakob disease, CJD, and other prion diseases. Since then, they have repeatedly improved and adapted it to detect other neurological diseases such as Parkinson's and dementia with Lewy bodies. The test, which already is used in clinical settings to diagnose sporadic CJD, is noted for its rapid and accurate results. Their latest findings could be a major advance for Alzheimer's diagnostics because the study points to tau seeds as potential biological markers for Alzheimer's disease in the brain. Scientists are testing therapies to slow the accumulation of tau clusters and the progression of neurological disease. In these efforts, they need more sensitive and accurate tests to better select clinical trial participants as assesses whether new therapeutical strategies work as hoped. Alzheimer's affects about 5.7 million people in the United States alone, at an estimated annual cost of $232 billion, according to the Alzheimer's Association. Today, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018. It is 7:22:20 seconds a.m. Eastern Standard Time. Science News. Threat of nightmare bacteria exhibiting resistance to last resort antibiotic colistin. Wide dissemination of colistin-resistant Escherichia coli, a growing therapeutical concern in rural communities in Vietnam revealed. Date December 20, 2018. Source Osaka University. Summary. Researchers examined the dissemination of colistin-resistant bacteria among residents of rural communities in Vietnam to find that the prevalence of colistin-resistant Escherichia coli in the intestines was extremely high, at about 70%. The team's research results were published in the Journal of Antimicrobial Chemotherapy. Colistin, an old antibiotic discovered in the 1950s, kills or inhibits the growth of gram-negative bacteria such as E. coli or Pseudomonas aeruginosa. It is one of the few critical antibacterial drugs for treating multi-drug-resistant bacteria and is listed on the WHO Essential Medicines List (EML) as a last-resort antibiotic. Colistin resistance is mostly related to chromosomal mutations. Although its frequency was low, a certain level of colistin resistance was observed. However, since colistin resistance caused by mutations was not transferred to other bacterial species, it was not considered problematic. However, a transmissible colistin resistance gene, MCR, was discovered in China in 2015, and the possibility of the transfer of colistin resistance to other bacterial species was shown, becoming a global concern. This presents a potential danger that carriage of MCR1 gene in pathogens with other resistant genes will produce superdrug-resistant bacteria called nightmare bacteria. This bacteria exhibits resistance to all antibiotics, even to the last resort antibiotic colistin, posing a great threat in infectious disease treatment. Drug-resistant strains are spread in livestock and livestock products in developing countries. This group has pointed out that meat contains colistin-resistant bacteria because a large amount of colistin has been added to livestock food in Vietnam. Thus, the group investigated the prevalence of colistin-resistant bacteria in Vietnam and revealed that the dissemination of colistin-resistant Escherichia coli, CRE, and the fecal microbiota of residents was extremely high, 70.4% of residents, and that the CRE had the transmissible colistin-resistant gene MCR. CRE carriers don't cause health problems immediately, but it is clear that the number of refractory infections for which antibiotics don't work will increase, which will become a great menace to clinical practice. In this borderless society, drug-resistant bacteria quickly spread beyond national and regional borders. So it is necessary to strengthen international surveillance systems and properly take preventive measures. Good day, this is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018 at 7:45 seconds a.m. Eastern Standard Time. AIDS and approach for targeting HIV reservoirs. Date December 20, 2018. Sources: Soup Pasteur. Summary: current HIV treatments need to be taken for life by those infected by antiretroviral therapy is unable to eliminate viral reservoirs lurking in immune cells. Scientists have identified the characteristics of CD4 T lymphocytes that are preferentially infected by the virus, thanks to metabolic activity inhibitors. The researchers have managed to destroy these infected cells or reservoirs ex vivo. The antiretroviral treatment used today is designed to block HIV infection but is not able to eliminate the virus from the body. The virus remains in the reservoirs, the CD4 T lymphocyte immune cells, the main targets of HIV. However, the virus does not infect all types of CD4 cell, and until now, the reason for this was not well known. In this study, scientists from the HIV Inflammation Persistence Unit at the Institut Pasteur and College have identified the characteristics of different CD4 subpopulations which are associated with HIV infection. The more the CD4 cells are differentiated or experienced, the more they need to produce energy to perform their function. Experiments have shown that it is the metabolic activity of the cell, and in particular its glucose consumption, that plays a key role in susceptibility to HIV infection. The virus primarily targets cells with high metabolic activity to multiply and hijacks the energy and products provided by the cell. This requirement constitutes a weakness for the virus and could be exploited to tackle infected cells. Scientists succeeded in blocking the infection ex vivo thanks to metabolic activity inhibitors that have already been investigated in cancer research. We have observed ex vivo that thanks to certain metabolic inhibitors, the virus is no longer able to infect cells and amplification is halted in reservoirs of patients receiving antiretroviral treatments. This is Professor Will, CBMD, PhD. Today is Friday, December 21st, 2018 at 7:33 seconds a.m. Eastern Standard Time. Science News. Stem cell-derived neurons stop seizures and improve cognitive function. People with untreatable epilepsy may one day have treatment. Date December 20, 2018. Source: Texas A&M University. Summary: about 3.4 million Americans, or 1.2% of the population, have active epilepsy. Although the majority respond to medication, between 20 and 40% of patients with epilepsy continue to have seizures even after trying multiple anti-seizure drugs. Even when the drugs do work, people may develop cognitive memory problems and depression, likely from the combination of the underlying seizure disorder and the drugs to treat it. The Central Texas Veterans Healthcare System is working on a better and permanent treatment for epilepsy. Their results published this week in the Proceedings of the National Academy of Science, PNAS. Seizures are caused when the excitatory neurons in the brain fire too much, and inhibitory neurons, the ones that tell the excitatory neurons to stop firing, aren't as abundant or aren't operating at their optimal level. The main inhibitory neurotransmitter in the brain is called GABA, short for gamma aminobutyric acid. Over the last decade, scientists have learned how to create induced pluripotent stem cells from ordinary adult cells, like a skin cell. These stem cells can then be coaxed to become virtually any type of cells in the body, inducing neurons that use GABA, called GABAergic interneurons. What we did is transplant human-induced pluripotent stem cells, derived GABAergic progenitor cells, into the hippocampus in the animal model of early temporal lobe epilepsy. The hippocampus is a region in the brain where seizures originate in temporal lobe epilepsy, which is also important for learning, memory, and mood. It worked very well to suppress seizures and even to improve cognitive and mood function in the chronic phase of epilepsy. Further testing showed that these transplanted human neurons form synapses or connections with the host excitatory neurons. They were also positive for GABA and other markers of specialized subclasses of inhibitory interneurons, which was the goal. Another fascinating aspect of this study is that the transplanted human GABAergic neurons were found to be directly involved in controlling seizures. As silencing the transplanted GABAergic neurons resulted in the increased number of seizures. One important aspect of the work is that the same cells can be obtained from a patient. This type of process, called autologous transplant, is patient-specific, meaning that there would be no risk of rejection of the new neurons, and the person would need anti-rejection medication. We will need to make sure that we are doing more good than harm. Going forward, we need to make sure that all the cells transplanted have turned into neurons, because putting indifferentiated pluripotent stem cells into the body could lead to tumors and other problems. The development of epilepsy often happens after a head injury, which is why the Department of Defense is interested in funding the development of better treatment and prevention options. A great deal of research is required before patients can be safely treated, but this publication shows a way in which patients can someday be treated with their own cells for the devastating effects of epilepsy, but perhaps also other diseases such as Parkinson's and Alzheimer's disease. Researchers caution that these tests were early interventions after the initial brain injury induced by status epilepticus, which is a state of continuous seizures lasting more than 5 minutes in humans. The next step is to see if similar transplants would work for cases for chronic epilepsy, particularly drug-resistant epilepsy. Currently, there is no effective treatment for drug-resistant epilepsy accompanied with depression, memory problems, and a death rate 5 to 10 times that of the general population. Our results suggest that induced pluripotent stem cell-derived GABAergic cell therapy has a promise for providing a long-lasting seizure control and relieving comorbidities associated with epilepsy. Today's is Professor Will, CBMD, PhD. Today's Friday, December 21st, 2018. It is 9:08:59 seconds Eastern Standard Time. Science News. Novel imaging technique brings diagnostic potential in an operating room. Date December 20, 2018. Source University of Illinois College of Engineering. Summary. Researchers have successfully visualized the tumor microenvironment of human breast tissue shortly after it was surgically removed from a patient in the operating room. The researchers achieved this by using a new portable optical imaging system. This work, which was reported in a paper published online December 19th in Science Advances, marks a major step toward providing cancer researchers with a new tool for tracking tumor progression and physicians new technology for tissue pathology and diagnostics. Typically, the process for diagnosing cancer takes several days. A surgeon first removes a tissue sample that is then processed with chemical dyes. Later, the sample is sent to a pathologist for examination and subsequent diagnosis. We believe that capturing the dynamic cellular and molecular features in freshly removed or biopsy tissue specimens contains valuable diagnostic and prognostic information that is currently lost when specimens are placed in a fixative and essentially killed quickly in order to preserve structure. Our imaging platform and methodology allow us to extract this new information in real time at the point of procedure. Portable optical imaging system uses precise light pulses to simultaneously image tissue in 4 modalities, enabling his team to study concurrent processes with cells and tissue that make up the tumor microenvironment. For example, collagen fibers appear in green, elastin fibers and flavin adenine dinucleotide containing cell cytoplasm appear in yellow, cell membranes, lipid boundaries, and extracellular vessels, EVs, appear in magenta, nicotinamide, adenine dinucleotide in cells, and lipids appear in cyan. The researchers were also interested in measuring tumor-related extracellular vessels, EVs, which are known to promote the spread of cancer. EVs do play an essential role in cancer progression. Quantifying EV densities may be developed as a potential biomarker for future cancer diagnosis. As a part of their studies, they also collected and imaged healthy breast tissue that surgeons had removed from cancer-free patients during breast reduction procedures. In a comparison of the two types of tissue, they found a clear difference in EV density between the cancerous and healthy tissue. For example, the cancerous tissue exhibited increased EV densities and had shorter tumor to margin distance. What we observed about the extracellular vessels is significant, but it could only be accurately determined with our new system. Noting how other portable optical imaging systems deployed in the operating room all alter the tissue samples either with fluorescent dyes or toxic ultraviolet light. Our imaging techniques works well with current cancer treatment routines and is free of any form of perturbation. The team's future plans include using the imaging system on tissue specimens obtained from needle biopsy procedures that are routinely performed in outpatient settings. They will also continue using the system on samples from the operating room. The imaging system is described in the paper. Intraoperative visualization of the tumor microenvironment and quantification of extracellular vessels by label-free nonlinear imaging, which was published online December 19, 2018. Today's is Professor Will, CBMD, PhD. Today's Friday, December 21st, 2018. It is 9:48:18 seconds a.m. Eastern Standard Time. Science News. Age is more than just a number. Machine learning may predict if you are in for a healthy old age. Researchers analyzed skin cells from more than 100 people of different ages to find molecular signatures that change as people get older. Date December 20th, 2018. Source Salk Institute. Summary. Researchers analyzed skin cells from the very young to the very old and looked for molecular signatures that can be predictive of age. By applying machine learning algorithms to these biomarkers, they were able to predict a person's actual age with less than 8 years error on average. For a study published December 19, 2018 in Genome Biology, a collaborative team of Salk Institute analyzed skin cells ranging from the very young to the very old and looked for molecular signatures that can be predictive of age. Developing a better understanding of the biological process of aging could eventually help to address health conditions that are more common in old age, such as heart disease and dementia. This experiment was designed to determine whether there are molecular signatures of aging across the entire range of human lifespan. We want to develop algorithms that can predict healthy aging and non-healthy aging and try to find the differences. The study provides a foundation for quantitatively addressing unsolved questions in human aging, such as the rate of aging during times of stress. The researchers focus on a type of skin cell called dermal fibroblasts, which generate connective tissue and help the skin to heal after injury. They chose this type of cell for two reasons. First, the cells are easy to obtain with a simple non-invasive skin biopsy. Second, earlier studies indicated that fibroblasts are likely to contain signatures of aging. This is because, unlike most types of cells that completely turn over every few weeks or months, a subset of these cells stays with us our entire lives. The investigators analyzed fibroblasts taken from 133 healthy individuals ranging in age from 1 to 94. To get a representative sample, the team studied an average of 13 people for each decade of age. The lab cultured the cells to multiply, then used a method called RNA sequencing. RNA-seq to look for biomarkers in the cells that change as people get older. RNA-seq uses deep sequencing technologies to determine which genes are turned on in certain cells, using custom machine learning algorithms to sort RNA-seq data. The team found certain biomarkers indicating aging and were able to predict a person's age with less than 8 years error on average. Rather than going into this research with an idea of what we wanted to find, we decided to look at the changes in expression of all the protein coding genes and let the algorithm sort it out. We used what's called an ensemble machine learning method to do this. The analysis from the Salk team was different from earlier approaches taken by other labs to study biological aging. Most previous studies focused on changes at only a few DNA methylation sites rather than looking at changes of expression on the whole genome. The dataset was also much larger than any research of this type that's ever been done before, because it included so many people representing a range of decades. The researchers have made the data public so that other investigators can use it. To validate the algorithm, the team also used fibroblasts from 10 patients with progeria, a genetic disease characterized by accelerated aging. Based on analysis of the molecular signature from these patients, who ranged in age from 2 to 8, the model predicted them to be about a decade older than their calendar age. The fact that our system can predict this kind of aging shows that this model is starting to get at the true underpinnings of biological age. Although this study uncovered age-related biomarkers, the investigators emphasized that just because something is predictive of aging doesn't mean it is the cause of aging. In the meantime, however, if the findings are validated, doctors could use this type of analysis to determine when to begin screening their patients for age-related conditions and to advise them about healthy lifestyle choices. More research needs to be done before any preventive treatments could be developed. One of the next steps of this research will look for these signatures in other types of cells. Aging is a driver of so many diseases, including Alzheimer's and other neurological problems. If we are able to show that the changes we've seen in fibroblasts are connected with aging in other types of cells, we may eventually be able to use these signatures to develop targeted interventions. Good day, this is Professor Will, CBMD PhD. Today's Friday, December 21st, 2018. It is 10:15, 42 seconds a.m. Eastern Standard Time. Science News. Bacteria rely on classic business model. Dates, December 20, 2018. Source, University of Basel. Summary. The pneumonia-causing pathogen, Pseudomonas aeruginosa, has developed a twin-track strategy to colonize its host. It generates two different cells, motile spreaders and virulent stickers. Researchers have now elucidated how the germ attaches to tissue within seconds and consecutively spreads just like a business model, settling, growing, expanding. The bacterium Pseudomonas aeruginosa is one of the most common and dangerous pathogens in hospitals, causing severe infections in patients, such as wound infections, pneumonia, and meningitis. During the first stage of infection, the pathogen needs to effectively attach to host tissue, but at the same time spread to distant sites. At later stages of infection, the pathogen needs to readjust its behavior to permanently colonize the host and hide from its immune system, for example, by forming protective biofilms. From spreading to adhering cells, the key to success for P. aeruginosa is its perfected colonization strategy during each division. The pathogen generates two distinct cell types, an attacking sticker and motile spreader. The balance between these two cell types is crucial for the pathogen in order to successfully dwell on tissues and to disseminate rapidly. In the initial phase of an infection, the spreader actively moves towards tissue surface of the host with a rotating cell propeller called flagellum. The researchers have now discovered that the flagellum also serves as a mechanical sensor. When the flagellum encounters a surface, bacteria rapidly respond by producing adhesive protrusions that firmly anchor the bacterium to the tissue. We were most surprised by how quickly this happens. Within seconds, the bacteria changes their program to stick to the surface. The tactile stimulus triggers the production of the bacteria signaling molecule cyclic di-GMP, which in turn initiates the formation of the cell protrusions. From adhering to spreading cells. Furthermore, the researchers have been able to show that upon division, surface-attached bacteria generate two different offspring. One daughter cell remains a sticker that can damage the underlying host tissue. The other one becomes a spreader, disseminating to distant sites. This process is also known as asymmetric cell division. This is due to the asymmetrical distribution of cyclic di-GMP in the dividing mother cell. While the daughter cell with high cyclic di-GMP level remains attached to the landing site, the cell with low cyclic di-GMP concentrations leaves the surface to colonize distant sites. Thus, P. aeruginosa exploits a simple business model: first establish, then grow, and finally expand. A smart tactic: conquer and hide. However, there is a limited window of opportunity for P. aeruginosa. After a few asymmetric divisions, the bacteria revert to dividing symmetrically, producing exclusively violent surface-attached offspring, thereby increasing the population at local sites exponentially. The pathogen has changed its strategy and is aiming for long-term establishment in the host and escaping the host defense systems. The researchers speculate that this colonization strategy is of general nature and can be found in a wide variety of bacteria that effectively colonize different surfaces such as stones, shower curtains, coffee cups, and human organs. Today's is Professor Will, CBMD, PhD. Today's Friday, December 21st, 2018 is 14:18:54 seconds Eastern Standard Time. Science News. Understanding repetitive head impacts in sports. Date December 20, 2018. Source Indiana University. Summary. A new study has found that eyeball and eyelid movement, or ocular motor function, which is used to detect symptomless brain injury, can be impaired by mild repetitive head impacts in football players, but the function may adapt mid-season even as athletes continue to incur head impacts. Repetitive subconcussive head impacts have quickly become one of the most complex public health issues. While various research finds insidious effects of repetitive subconcussive head impacts, we found unique ocular motor response to head impacts and realized that we still have much more to learn about brain response to trauma. Head injuries in sports remain a focus of researchers throughout the world. Much of this work focuses on understanding repetitive subconcussive head impacts, hits that don't meet the criteria for concussive diagnosis but still may have long-term effects. The goal is to identify a safe threshold for subconcussive brain injury that does not necessarily trigger immediate symptoms such as headache, dizziness, and disorientation. Research measured the near point of convergence, the measurement of the closest point of focus before double vision occurs, in more than a dozen high school football players over one season. Near point of convergence was measured 14 times the baseline measurement, a pre- and post-game measurement during 6 in-season games and post-season follow-up. Players experienced more than 8,000 recorded hits over this period, and the researchers found a significant increase up to 33% in near point of convergence resulting from subconcussive head impact frequency up to the middle of the football season. However, near point of convergence began to move back toward the baseline measurement from mid-season on, even though players continued to experience subconcussive head impacts. Based on our previous studies, we thought that near point of convergence would be impaired throughout the entire season. Instead, near point of convergence was normalized to baseline by the last quarter of the season, when players play their hardest to make it to the playoff stage. The research concluded that while near point of convergence is one of the most prevalent clinical assessments for concussions, further study is necessary to determine whether it is the best tool in tracking long-term subconcussive brain damage. Researchers are planning a large-scale longitudinal study to further investigate the use of ocular motor function testing on repetitive subconcussive head impacts. Our study further lays the groundwork for understanding the usefulness of limitations of convergence as a clinical biomarker for understanding acute and chronic subconcussion. When evaluating the severity of brain damage, it is important to assess not only ocular motor function, but also other neurological variables, including fluid and biomarkers, and neuroimaging, and the ultimate goal of brain injury prevention. Good day, this is Professor Will, CBMD, PhD. Today's Friday, December 21st, 2018 is 14:28:34 seconds Eastern Standard Time. Science News. Impact of diet intervention in autoimmunity in mice. Date December 20th, 2018. Source Yale University. Summary. Could a change in diet be beneficial to people with autoimmune disease such as lupus? Researchers have revealed how a dietary intervention can help prevent the development of this autoimmune disease in susceptible mice. They first identified a single bacterium, Lactobacillus reuteri, in the gut of the mice that triggered an immune response leading to the disease. Specifically, in lupus-prone mice, L-reuteri stimulated immune cells known as dendritic cells, as well as immune system pathways that exacerbated disease development. Researchers feeding mice resistant starch, a diet that mimics a high-fiber diet in humans. Resistant starch is not absorbed in the small intestine, but ferments in the large intestine, enriching good bacteria and causing the secretion of short-chain fatty acids. This in turn suppresses both the growth and movement of L-reuteri bacteria outside the gut that would otherwise lead to autoimmune disease. While more research is needed to discern how the findings translate to humans, the study details an important link between diet, gut bacteria, and autoimmunity. We dissected molecularly how diets can work on the gut microbiome. We identified a pathway that is driving autoimmune disease and mitigated by the diet. The study also found an imbalance of gut microbes in a subset of lupus patients that was similar to what they observed in lupus-prone mice, not given the starch diet. In this subject of lupus patients, the high-fiber diet could potentially be beneficial to prevent or ameliorate the condition, in addition to other diseases that activate the same immune pathway. It may have implications beyond lupus. Today's is Professor Will, CBMD PhD. Today is Friday, December 21st, 2018. It is 14:44:55 seconds Eastern Standard Time. Science News. Virologists discover safer potential Zika vaccine. New mechanism could be huge leap for immunology. Date December 20, 2018. Source University of Nebraska-Lincoln. Summary in mouse trials of vaccine based on recombinant adenovirus protected against Zika without evidence of antibodies. Reports that shown Zika antibodies could worsen dengue virus infection. Many reports show that antibodies against Zika virus can worsen dengue virus infection, which, like Zika, is caused by a mosquito-borne virus. This phenomenon is referred to as antibody-dependent enhancement, ADE, of disease. This has been an obstacle to the development of effective and safe dengue virus vaccines. If you have immunity to one of these viruses and get infected by a second one, the illness can be much worse. The body makes the wrong immune response. Researchers have been studying potential Zika vaccines since 2016, shortly after a Zika outbreak in Brazil that was declared a global public health emergency by the World Health Organization. First discovered in Uganda's Zika forest in 1947, the virus initially was believed to cause only mild or asymptomatic infection in humans. However, outbreaks in Brazil in 2015 and 2016 resulted in higher than historical rates of congenital birth defects in babies born in infected mothers and an increase in neurological disorders, Guillain-Barré syndrome in adults. In 2016, between 500,000 and 1.5 million suspected cases of Zika infection were reported worldwide, with 4,300 related cases of microcephaly, or abnormally small heads in infants. Researchers developed a genetically altered version of adenovirus. Scientists used two forms of attenuated adenovirus to serve as vectors to deliver the Zika vaccine. Adenoviruses, which typically cause mild illness such as common cold, are modified so that they are replication defective and incapable of causing disease. The modified or attenuated viruses are regarded as safe and highly effective vaccine vectors capable of inducing long-lasting protective immune responses against infectious pathogens. They inserted the PRM-E structural genes of Zika into key areas of adenovirus type 4 and adenovirus type 5 genomes. Tested in mice, both vaccines offered strong T responses, substantial protection against Zika infection. However, the vaccine based on type 4 adenovirus induced strong T cell response with undetectable antibody. T cells are a type of white blood cell that are at the core of the system that tailors the body's adaptive immune response to specific pathogens. To our knowledge, this is the first reported vaccine that uses the PRM-E gene of Zika virus to induce protective immunity without inducing anti-Zika virus antibodies. The lack of antibodies may very well circumvent the potential risk of ADE, resulting in an effective and safer vaccine than those currently in clinical trials. More studies are needed in order to determine why one virus vector had different results than the other. We hope that we can produce a safer vaccine, researchers say. If we can figure out the mechanism, we might be able to apply it to other vaccine strategies.